Good evening. In fact, a very good morning. It's early morning, 1:45, September 3rd. I am Dr. Bobby Jacob, KORAH, and on behalf of New Age Cosmetic White EdTech Healthcare Consultancy, I'd like to welcome all of you to the continuous lectures of for those aspiring to apply for the Neet exams, All India Institute of Medical Sciences, and the Council for the Scientific Industrial Research exams on the National Eligibility Test, and the Neet test, National Eligibility Test for the medical and dental graduates too. So continuing with the lectures as of yesterday, the last topic we dealt with yesterday was the Michaelis-Menten Model accounts for the kinetic properties of many enzymes. So the primary function of enzymes is to enhance rates of reaction so that they're compatible with the needs of the organism. To understand how enzymes function, we need to have a know of the kinetic descriptions of the activities. But while I continue with the lectures, just a point to mention is that the starting topic of each lecture would be the ending subject of the previous lecture, so that there's a point of continuity on where you stood and where we last stopped, right? So this is how the continuation goes. And make your notes on screenshots and take printouts and make notes. And as usual, connect with me if you have any doubts. Okay, continuing with the subject. To understand how enzymes function, we need a kinetic description of the activity. For many enzymes, the rate of catalysis, VO, which is defined as the number of moles of product formed per second, varies with the substrate concentration S. The rate of catalysis rises linearly as substrate concentration increases and then begins to level off and approach a maximum at higher substrate concentrations. The graph shows that. Before we can accurately interpret this graph, we need to understand how it is in fact generated. So consider an enzyme that catalyzes the S to P by the following pathway, where enzyme plus substrate gives enzyme substrate to enzyme plus to P, E plus P by the formula as mentioned here. And from a typical Michaelis-Menten plot, because in fact, before we could continue on that, let's make a mention of the Michaelis-Menten equation. So where V0 is equal to Vmax S bar S plus Km. The meaning of Km is evident from the equation of equation number 23. When S is equal to Km, then V0 is equal to Vmax by 2. This Km is equal to the substrate concentration, of which the reaction rate is half its maximum value. Km is an important important characteristic of an enzyme-catalyzed reaction and is significant for its biological function. The significance of Km and Vmax values. The Michaelis constant Km and the maximal value rate Vmax can be readily derived from rates of catalysis measured at a variety of substrate concentrations. If any enzyme operates according to a simple scheme given in equation 23, the derivation of Km and Vmax is most commonly achieved with the use of curve-fitting programs. On a program, see the appendix to this chapter later for an alternate means of determining Km and Vmax. The Km values of enzymes range widely. For most enzymes, Km lies between 10 raised to minus 1 and 10 raised to minus 7 m. The Km value for an enzyme depends on the particular substrate and on environmental conditions such as pH, temperature, and ionic strength. The Michaelis constant Km has two meanings. First, Km is the concentration of substrate of which half the active sites are filled. This K provides a measure of the substrate concentration required for significant catalysis to occur. In fact, for many reasons. For many enzymes, experimental evidence suggests that Km provides an approximation of substrate concentration in vivo. When the Km is known, the fraction of sites filled, FES at any substrate concentration can be calculated from. Now, from the formula for that is FES, which is equal to V by Vmax is equal to S by S plus Km. Second, Km is related to the rate constants of the individual steps in the catalytic scheme. Km is defined as K minus 1 plus K2 by K1. Consider a limiting case in which K2 is much greater than K2. Under such circumstances, the ES complex dissociates to E and S much more rapidly than product is formed. Under these conditions, K minus 1 is much greater than K2. So the dissociation constants of the ES complex is given by Km is equal to K minus 1 by K1. And Kes is equal to E into S by Es is equal to K minus 1 by K1. So, in other words, Km is equal to the dissociation constant of the ES complex of K2 is much smaller than K1. When this condition is met, Km is a measure of the strength of the ES complex. A high Km indicates weak binding, a low Km indicates strong binding. It must be stressed that Km indicates the affinity of the ES complex only when K minus 1 is much greater than K2. The maximal rate Vmax reveals the turnover number of an enzyme, which is the number of substrate molecules converted into product by an enzyme molecule in a unit time when the enzyme is fully saturated with the substrate. It is equal to the kinetic constant K2, which is also called Kcat. The maximal rate Vmax reveals the turnover number of an enzyme if the concentration of active salts ET is known. Because K2 is equal to Vmax by ET, Vmax is equal to K2 by ET. For example, a 10 raised to minus 6 m solution of carbonic anhydrous catalyzes the formation of 0.6 m H2CO3 per second when it is fully saturated with substrate. Hence, K2 is 6 into 10 raised to 5 s minus 1. This turnover number is one of the largest known. Each catalyzed reaction takes place in a time equal to 1 by K2, which is 1.7 micro s for carbonic anhydrase. The turnover number of most enzymes with their physiological substrates fall in the range from 1 to 10 raised to 4 per second. Vmax and Km can be determined by double reciprocal plots. Before the availability of computers, the determination of Km and Vmax values required algebraic manipulation of the basic Michaelis-Menten equation. Because Vmax is approached asymptomatically, it is impossible to obtain a definitive value. Right? So, from a typical Michaelis-Menten plot, because Km is the concentration of substrate at Vmax by 2, it is likewise impossible to determine the accurate value of Km. Right? However, Vmax can be accurately determined if the Michaelis-Menten equation is transformed into one that gives a straight line plot. Taking the reciprocal of both sides of equation 23 gives 1 by Vo is equal to 1 by Vmax plus Km by Vmax plus into 1 by S. A plot of 1 by V0 versus 1 by S called a Lineweaver-Burk or double reciprocal plot yields a straight line with an intercept of 1 by Vmax and a slope of Km by Vmax figure. The intercept on the x-axis is minus 1 by Km. Right? Double reciprocal plots are especially useful for distinguishing between competitive and non-competitive inhibitors. In competitive inhibition, the intercept on the y-axis of the plot of 1 by V0 versus 1 by S is the same in the presence and in the absence of inhibitor, although the slope is increased. Right? So that the intercept is unchanged is because a competitive inhibitor does not alter Vmax at a sufficiently high concentration. Virtually all the active sites are filled by substrate, and the enzyme is fully operative. The increase in the slope of the 1 by V0 versus 1 by S plot indicates that the strength of the binding of competitive inhibitor. In the presence of competitive inhibitor, equation 31 is replaced by 1 by V0 is equal to 1 by Vmax plus Km by Vmax into 1 plus 1 by K1 into 1 by S, in which I is the concentration of inhibitor and K1 is the dissociation constant of the enzyme to inhibitor complex, enzyme inhibitor complex, where E plus 1 is equal to EI, E plus I is equal to EI, K1 is equal to EI by EI. In other words, the slope of the plot is increased by the factor 1 plus 1 by K1 in the presence of competitive inhibitor. In non-competitive inhibition, the inhibitor can combine with either the enzyme or the enzyme substrate complex. In pure non-competitive inhibition, the values of the dissociation constants of the inhibitor and enzyme and of the inhibitor and enzyme substrate complex are equal. The value of Vmax is decreased to a new value called Vmax, and so the intercept of the vertical axis is increased. The new slope, which is equal to Km by Vmax, is larger by the same factor. In contrast with Vmax, Km is not affected by pure non-competitive inhibition. The maximal velocity in the presence of a pure non-competitive inhibitor, Vmax, is given by Vmax is equal to Vmax by 1 plus I by Ki. Right, that's the topic there. The next topic will be F8 on allostericc enzymes. The Michaelis-Menten model has greatly assisted the development of enzyme chemistry. Its virtues are simplicity and broad applicability. However, the Michaelis-Menten model cannot account for the kinetic properties of many enzymes. An enzyme group of enzymes that do not obey Michaelis-Menten kinetics comprises the allostericc enzymes. These enzymes consist of multiple subunits and the multiple active sites. The allostericc enzymes have an allostericc site in addition to the active site. Allostericc is derived from the Greek word allo and steric. Allo means other, steric means tight. An effective molecule can bind with this site and can either activate or deactivate the enzyme molecule. The effective molecule is termed as modulator, regulator, or ligand. The effective molecule may be either activators or inhibitors. The activators are generally one of the substrate or cofactors in most cases. The allostericc inhibitors are the end products of the metabolic pathways in which particular enzyme is participating. Thus, this kind of enzyme inhibition is also called as feedback inhibition or end product inhibition or retro inhibition. The effect of allosteric is reversible. When they are withdrawn, the enzyme resumes its original activity, and the graph shows this. Allosteric enzymes often display sigmoidal plots of the reaction velocity V0 versus substrate concentration S rather than the hyperbolic plots predicted by the Michaelis-Menten equation. Have a look at this and you'll get to know that. Right? The sigmoid curve. Hyperbolum is just a full thing, a full curve, going way up. The two models explain the kinetic behavior of allostericc enzymes. I hope you got that part. Hyperbole would be just like that, a full thing. Here it goes down and then up. So that's a sigmoid curve. Right? Right, so two models explain the kinetic behavior of allostericc enzymes. The sigmoidal dependence of V0 on S reflects subunit cooperatively and has inspired two models to explain these cooperative interactions. And the first model, the symmetry model, proposed by Jacques Monod and colleagues in 1965, an allostericc enzyme can exist in only two conformations, active and inactive. Figure A, all subunits are in the active form or all are inactive. Every substrate molecule that binds increases the probability of a transition from the inactive to the active state. And in continuation of that, we get to see that in the second model, the sequential model, figure B, proposed by Koshler in 1966, there are still two conformations, but subunits can undergo the conformational change and individually. Binding of substrate increases the probability of conformational change. A conformational change in one subunit makes a similar change in an adjacent subunit, as well as the binding of a second substrate molecule more likely. There are more potential intermediate sites in this model than in the symmetry model. The two models are not mutually exclusive. The symmetry model may be viewed as the all or none limiting case of the sequential model. The precise mechanism of allosteric interaction has not been established. Different allosteric enzymes may have different mechanisms for cooperative interactions. In allosteric enzymes, the binding of substrate to one active site can affect the properties of other active sites in the same enzyme molecule. A possible outcome of this interaction between subunits is that the binding of substrate becomes cooperative. That is, the binding of substrate to one active site of the enzyme facilitates substrate binding to the other active site or sites. Such cooperativity results in a sigmoidal plot of V0 versus S. In addition, the activity of an allosteric enzyme may be altered by regulatory molecules that are reversibly bound to specific sites other than the catalytic sites. The catalytic properties of allosteric enzymes can thus be adjusted to meet the immediate needs of a cell. For this reason, allosteric enzymes are key regulators of metabolic pathways in the cell. For example, in threonine metabolism pathway, the end product, isoleucine, acts as inhibitor and binds to enzyme of first step threonine deaminase. Aspartate transcarbamylase, or let me say that again, aspartate transcarbamylase, ATCase, one of the best understood allosteric enzymes, catalyze the synthesis of N-carbamyl aspartate and the first intermediate in the synthesis of pyrimidines. ATCase is feedback inhibited by cytidine triphosphate CTP. The final product of the pathway, ATP reverses this inhibition. ATCase consists of separable catalytic C3 subunits, which bind to substrates, and regulatory R2 subunits, which bind CTP and ATP. The inhibitory effect of CTP, the stimulatory action of ATP, and cooperative binding of substrates are mediated by the large changes in quaternary structure on binding substrates. The C3 subunits of the C6R6 enzyme move apart and reorient themselves. Positive cooperativity. Positive cooperativity means that the enzyme is much more sensitive to changes in the amount of substrate available to it. If you consider a situation such as a glucose breakdown, you'll really realize that the amount of glucose being degraded for energy but very greatly vary with the physiological situation of the cell. If it needs a great deal of energy, it will be degrading a lot of glucose. If not, it will be degrading very little or none at all. The enzymes in the breakdown pathway would therefore have to cope with the big differences in the amount of substrate that they have to convert, and the greater sensitivity to substrate concentration produced by a positive substrate cooperativity will be of great assistance to this. Thus, in positive cooperativity, binding of substrate to any one active site increases the chance of binding substrate to other sites. Negative cooperativity. This means that an enzyme is much less sensitive to substrate change and almost becomes independent of the concentration of substrate. This may be useful with a substrate such as coenzyme, perhaps whose concentration may change due to reactions in the cell which are not directly related to the pathway in which our enzyme is involved. It may well be an advantage in these circumstances if the enzyme does not react to changes in that substrate. Here, binding of substrate to any one subunit decreases the affinity of enzyme for other substrates. Going to the next topic, F9 enzymes can be inhibited by specific molecules, inhibitors. The activity of many enzymes can be inhibited by the binding of specific molecules, small molecules and ions. This means inhibiting enzyme activity serves as a major control mechanism in biological systems. Right, and this means inhibiting enzyme activity serves as a major control mechanism in biological systems. The regulation of allosteric enzymes typifies this type of control. In addition, many drugs and toxic agents act by inhibiting enzymes. Inhibition by particular chemicals can be a source of insight into the mechanism of enzyme action. Specific inhibitors can often be used to identify residues critical for catalysis. the value of transition state analogs as potent inhibitors will be discussed from here on or shortly. Enzyme inhibition can either be reversible or irreversible. An irreversible inhibitor dissociates very slowly from its target enzyme because it has come tightly bound to the enzyme either covalently or non-covalently. Some irreversible inhibitors are important drugs. Penicillin acts by covalently modifying the enzyme transpeptidase, thereby preventing the synthesis of bacterial cell walls and thus killing the bacteria. Aspirin acts by covalently modifying the enzyme cyclooxygenase, reducing the synthesis of inflammatory signals. Reversible inhibition, in contrast with irreversible inhibition, is characterized by a rapid dissociation of the enzyme inhibitor complex. In competitive inhibition, an enzyme can bind substrate forming ES complex and ES complex, or inhibitor E1 but not both ESI. The enzyme substrate inhibitor. The competitive inhibitor resembles the substrate and binds to the active site of the enzyme. The substrate is thereby prevented from binding to the same active site. A competitive inhibitor diminishes the rate of catalysis by reducing the proportion of enzyme molecules bound to a substrate at any given inhibitor's concentration. Competitive inhibition can be relieved by increasing the substrate concentration. Under these conditions, a substrate outcompetes the inhibitor for the active site. Methotrexate is a structural analog of tetrahydrofolate, a coenzyme for the enzyme dihydrofolate reductase, which plays a role in the biosynthesis of purines and pyrimidines. It binds to dihydrofolate reductase 1,000-fold more tightly than the natural substrate and inhibits nucleotide-based synthesis. It is used to treat cancer. So many antibacterial drugs work on principle of competitive inhibition. For example, sulfur drugs are believed to be competitive inhibitor of enzyme metabolizing p-aminobenzoic acid PABA or PABA, which is essential vitamin or vitamin actin as coenzyme for many bacterial enzymatic reactions and structurally similar to sulfanilamide, a sulfur drug. Similarly, malonate is structurally similar to succinate, the substrate for pyruvate dehydrogenase, and thus competes for enzyme. In non-competitive inhibition, which is also reversible, the inhibitor and substrate can bind simultaneously to the enzyme molecule at different binding sites. A non-competitive inhibitor acts by decreasing the turnover number rather than by diminishing the proportion of enzyme molecules that are bound to substrate. Non-competitive inhibition, in contrast with the competitive inhibition, cannot be overcome by increasing the substrate concentration. Such type of inhibition is shown by allosteric enzymes, which are regulated by a feedback mechanism. A more complex pattern called mixed inhibition is produced when a single inhibitor both hinders the binding of substrate and decreases the turnover number of the enzyme. Competitive and non-competitive inhibition are kinetically distinguishable is the first point here. How can we determine whether a reversible inhibitor acts by a competitive or non-competitive inhibition? Let us consider only enzymes that exhibit Michaelis-Menten kinetics. Measures of the rates of catalysis at different concentrations, substrate and inhibitor, serve to distinguish the two types of inhibition. In competitive inhibition, the inhibitor completes with the substrate for the active site. The dissociation constant for the inhibitor is given by Ki is equal to E into I by Ei. Because increasing amount of substrate can overcome inhibition, Vmax can be attained in the presence of a competitive inhibitor. The hallmark of competitive inhibition is that it can be overcome by a sufficiently high concentration of a substrate. However, the apparent value of Km is altered. The effect of a competitive inhibitor is to increase the apparent value of Km. This new value of Km, called Kap-M, is numerically equal to Kap-M is equal to Km into 1 plus 1 by Ki, where I is the concentration of inhibitor and Ki is the dissociation constant for the enzyme inhibitor complex. As the value of I increases, the value of Kap-M increases. In the presence of a competitive inhibitor, an enzyme will have the same Vmax as in the absence of an inhibitor. In non-competitive inhibition, substrate can bind to the enzyme inhibitor complex. However, the enzyme inhibitor substrate complex does not proceed to form product. The value of Vmax is decreased to a new value called Vmax, while the value of Km is unchanged. Why is Vmax lowered while Km remains unchanged? In a sense, the inhibitor simply lowers the concentration of functional enzyme. The remaining enzyme behaves like a more dilute solution of enzyme. Vmax is lower, but Km is the same. Non-competitive inhibition cannot be overcome by increasing the substrate concentration. Going on to the next topic is section F10, catalytic strategies. A few basic catalytic principles that are used by many enzymes. Enzymatic catalysis begins the substrate binding. The binding energy is the free energy released in the formation of a large number of weak interactions between the enzyme and the substrate. We can envision this binding energy as serving two purposes. It establishes substrate specificity and increases catalytic efficiency. Only the correct substrate can participate in most or all of the interactions with the enzyme, and thus maximizing binding energy, accounting for the exquisite substrate specificity exhibited by many enzymes. Furthermore, the full complement of such interactions is formed when the substrate is in this transition stage. Thus, interactions between the enzymes and the substrate not only favor substrate binding but stabilize the transition stage, thereby lowering the activation energy. The binding energy can also promote structural changes in both the enzyme and the substrate that facilitate catalysis, a process referred to as induced fit. Enzymes commonly employ one or more of the following strategies to catalyze specific reactions. 1. Covalent catalysis. The active site contains a reactive group, usually a power nucleophile, that becomes temporarily covalent modified in the course. Of catalysis, the proteolytic enzyme chymotrypsin provides an excellent example of this mechanism. 2. General acid-base catalysis. In general acid-base catalysis, a molecule other than water plays the role of a donor or acceptor. Chymotrypsin uses a histidine residue as a base catalyst to enhance the nucleophile power of serine. And metal ion catalysis. Metal ions can function catalytically in several ways. For instance, a metal ion may serve as an electrophilic catalyst, stabilizing negative change on a reaction intermediate. Alternatively, the metal ion may generate a nucleophile by increasing the acidity of a nearby molecule, such as water, in the hydration of carbon dioxide by carbonic anhydrase. Finally, the metal ion may bind to substrate, increasing the number of interactions with the enzyme and thus the binding energy. This strategy is used by NMP kinases. Catalysis by approximation. Many reactions include two distinct substrates. In such cases, the reaction rate may be considerably enhanced by bringing the two substrates together along a single binding surface of an enzyme. NMP kinases bring two nucleotides together to facilitate the transfer of phosphorus group from one nucleotide to the other. And the next section will be F11, regulation of enzymatic activity. The activity of proteins includes enzymes most often must be regulated so that they function at the proper time. Starting with the topic of catalytic strategies. Enzyme catalysis begins with the substrate binding. The binding energy is the free energy released in the formation of a large number of weak interactions between the enzyme and the substrate. We can envision this binding energy as serving two purposes. It establishes substrate specificity and increases catalytic efficiency. Only the correct substrate can participate in most or all of these interactions with the enzyme and thus maximizing binding energy, accounting for the exquisite substrate specificity exhibited by many enzymes. Furthermore, the complement of such interactions is formed only when the substrate is in the transition stage. Thus, interactions between the enzyme and the substrate not only favor substrate binding but stabilize the transition stage, thereby lowering the activation energy. The binding energy can also promote structural changes in both the enzymes and the substrate that facilitate catalysis, a process referred to as induced fit. Enzymes commonly employ one or more of the following strategies to catalyze specific reactions. Covalent catalysis. In covalent catalysis, the active site contains a reactive group, usually a powerful nucleophile that becomes temporarily covalent, modified in the course of catalysis. The proteolytic enzyme chymotrypsin provides an excellent example of this mechanism. General acid-base catalysis. In general acid-base catalysis, a molecule other than the water plays the role of a proton donor or acceptor. Chymotrypsin uses a histidine residue as a base catalyst to enhance the nucleophile power of serine. Metal ion catalysis. Metal ions can function catalytically in several ways. For instance, a metal ion may serve as an electrophilic catalyst, stabilizing a negative charge on a reaction intermediate. Alternatively, the metal ion may generate a nucleophile by increasing the acidity of a nearby molecule, such as water and the hydration of CO2 by carbonic anhydrase. Finally, the metal ion may bind to substrate, increasing the number of interactions with the enzyme and thus the binding energy. This strategy is used by NMP kinases. And catalysis by approximation. Many reactions include two distinct substrates. In such cases, the reaction rate may be considerably enhanced by bringing the two substrates together along a single binding surface of an enzyme. NMP kinases bring two nucleotides together to facilitate the transfer of a phosphoryl group of one nucleotide. We are going on to the next topic, F11, where we deal with the regulation of enzymic activity. The activity of proteins including enzymes, often must be regulated so that they function at the proper time and place. The biological activity of proteins is regulated by four principal ways. One is the allosterically control, where proteins contain distinct regulatory sites and multiple functional sites. Regulation of small signal molecules is a significant means of controlling the activity of many proteins. The binding of these regulatory molecules at sites from the active site triggers conformational changes that are transmitted to the active site. Many allosteric proteins show the property of cooperative activity at a functional site affects the activity at others. And while proteins as a consequence, a slight change in substrate concentration can produce substantial changes in the activity, and proteins displaying allosteric control are thus information transducers. The activity can be modified in response to signal molecules or to information shared among active sites. The two best understood allosteric proteins are the enzyme aspartate transcarbamylase, ATCase, and the oxygen-carrying protein hemoglobin. Catalysis by aspartate transcarbamylase of the first step in pyrimidine biosynthesis is inhibited by cytidine triphosphate. The final product of that biosynthesis is an example of feedback inhibition. The binding of O2 by hemoglobin is cooperative and is regulated by H+, CO2, and 2,3-biphosphoglycerate 2,3-BPG. Multiple forms of enzymes. Isozymes or isoenzymes provide an avenue for varying regulation of the same reaction at distinct locations or times. Isoenzymes are homologous enzymes within a single organism that catalyze the same reaction but differ slightly in the structure and more obviously in Km and Vmax values as well as regulatory properties. Often isoenzymes are expressed in a distinct tissue or organelle or at a distinct stage of development. Reversible covalent modification. The catalytic properties of many enzymes are markedly altered by the covalent attachment of a modifying group. Most commonly, a phosphoryl group, ATP, serves as a phosphoryl donor in these reactions, which are catalyzed by protein kinases. The removal of phosphoryl groups by hydrolysis is catalyzed by protein phosphatases. Cyclic AMP serves as an intracellular messenger in the transduction of many hormonal and sensory stimuli. Cyclic AMP switches on protein kinase, a pKa multifunctional kinase by binding to the regulatory subunit of the enzyme. They were released in the active catalytic subunits of pKa, phenylketonuria acid. In the absence of CAMP, the catalytic sites of pKa are occupied with pseudo-substrate sequences of the regulatory subunit. Proteolytic activation. The enzymes controlled by some of these mechanisms cycle between active and inactive states. A different regulatory motif is used to irreversibly convert an inactive enzyme into an active one. Many enzymes are activated by the hydrolysis of a few or even one peptide bond in inactive precursors called zymogens or proenzymes. This regulatory mechanism generates digestive enzymes such as chymotrypsin, trypsin, and pepsin, caspases, which are proteolytic enzymes that are the executioners in programmed cell death, or apoptosis are proteolytically activated from the procaspase form. Right. And blood clotting is due to a remarkable cascade of zymogen activations. Active digestive and clotting enzymes are switched off by the irreversible binding of specific inhibitory proteins that are irresistible lures to other molecular prey. Going on to the next topic, F12, isoenzymes, abenzymes, and riboenzymes. Isoenzymes or isoenzymes, enzymes that differ in amino acid sequence yet catalyze the same reaction. Usually these enzymes display different kinetic parameters, which such as Km or different regulatory properties. They are encoded by different genetic loci, which usually arise through gene duplication and divergence. Isozymes differ from allozymes, which are enzymes that arise from allelic variation at one gene locus. Isozymes can often be distinguished from one another by biochemical properties such as electrophoric mobility, as they differ from each other in electric charges. The existence of isozymes permits the fine-tuning of metabolism to meet the particular needs of a given tissue of developmental stage. Consider the example of lactate dehydrogenase LDH, an enzyme that functions in anaerobic glucose metabolism and glucose synthesis. Two human beings have two isozymic polypeptide chains for this enzyme. The H isozyme highly expressed in heart and the M isozyme found in skeletal muscle. The amino acid sequences are 75% identical. The functional enzyme is tetrameric and many different combinations of subunits are possible. The H4 isozyme found in the heart has a higher affinity for substrates than does the M4 isozyme. The two isozymes also differ in that high level of high pyruvate allosterically inhibit the H4 but not the M4 isozyme. The other combinations such as H3M have intermediate properties depending on the ratio of the two types or two kinds of chains. Isoenzymes are useful in separation of taxa, that are chemotaxonomic studies and catalysis of reversible reaction. One form of enzyme can catalyze a reaction in one direction, while other form of the same enzyme can catalyze a reaction in the opposite direction. Abzyme. In 1969, William Jenks proposed the antibodies specific for the transition state of a chemical reaction should have catalytic power. This incisive prediction was realized in 1986 when the laboratories of Richard Lerner and Peter Schultz found that the catalytic antibodies could be produced by using transition state analogues as immunogens. The preparation of an antibody that catalyzes the insertion of a metal ion into a porphyrin nicely illustrates this experimental approach. Ferrochelatase, the final enzyme of biosynthetic pathway for the production of heme, catalyzes the insertion of Fe2 plus into porphyrin 9. The nearly planar porphyrin must be bent for ion to enter. N-methylprotoporphyrin is a potent inhibitor of ferrochelatase. This compound resembles a transition stage because N-alkylation forces the porphyrin to bend. Moreover, it was known that N-alkyl porphyrin chelate mental ion are 104 times than the unalkylated counterparts. Bending increases the exposure of lone pairs of electrons of the pyrrole nitrogen to solvent, which facilitates chelation. Indeed, an effective catalyst was produced using an N-alkyl porphyrin as the iminogen. The resulting antibody distorted a planar porphyrin to facilitate the entry of a metal ion. 80 porphyrin molecules were methylated per hour antibody molecule, a rate only tenfold less than that of the ferrochelatase. The uncatalyzed reaction was 2,500 times as slow as the antibody-catalyzed reaction. And antibodies catalyzing many other kinds of chemical reaction, exemplified by ester and amide hydrolysis, amide bond formation, transesterification, photoinduced cleavage, photoinduced dimerization, decarboxylation, and oxidation have been using similar strategies. The power of transition state analog is now evident since they provide evidence insight into catalytic mechanism. They can serve as potent and specific inhibitors of enzymes. They can be used as immunogens to generate a wide range of novel catalysts. Ribozyme. Thomas Sheck in 1983 found that the precursor of a ribosomal RNA in Tetrahymena, a ciliated protozoan, undergoes self-splicing. The intron in this precursor RNA molecule is precisely removed by catalytic activity of RNA itself. This liberated intron then loses a short 5' terminal sequence to form a 385 nucleotide RNA molecule that catalyzes transformation of other RNA molecules. The intron-derived RNA catalyzes the cleavage of joining RNA chains at specific site without itself being consumed. Hence, it is true enzyme ribozyme, another enzyme that contains a key RNA component, is ribonuclease P, RNase P, which catalyzes the maturation of tRNA by removing nucleotides from the 5' end of the precursor molecule. In Tetrahymena, a ciliated protozoan, a 440 nucleotide intron is removed from a 6.4 kb precursor to yield the mature 26S RNA molecule. In an elegant series of studies of this splicing reaction, Thomas Sheck and his co-workers established that the RNA spliced itself to precisely excise the 440 nucleotide intron. The self-splicing reaction requires an added guanosine nucleotide as an attacking group that becomes transiently incorporated into the RNA. G binds the RNA and then attacks the 5' site to form a phosphodiester bond with the 5' end of the intron. I think that's in prime. Okay. This transesterification reaction generates a 3-OH group at the end of the upstream exon. This newly attached 3-OH group then attacks the 3' splice. This second transesterification reaction joins the two exons and leads to the release of the 440 nucleotide intron. A phosphodiester bond is formed between the two exons. Like catalysis by protein enzymes, self-catalysis of bond formation breakage of this RNA precursor is highly specific. Messenger RNA precursors in the mitochondria of yeast and fungi also undergo self-splicing, as to do some RNA precursors in the chloroplasts of unicellular organisms such as Chlamydomonas. Self-splicing reactions can be classified according to the nature of the unit that attacks the upstream splice site. Group 1 self-splicing is mediated by a guanosine cofactor as in Tetrahymena. The attacking moiety in Group 2 splicing is a 2-OH group of a specific adenylate. The next topic will be G1, the conformation of proteins and polypeptides, right? So secondary structure in 51, Linus Pauling and Robert Corey proposed two periodic structures called the alpha helix on the beta pleated sheet. Subsequently, other structures such as the reverse turn and omega loop were identified. The alpha helix is a rod-like structure. A tightly coiled backbone forms the inner part of the rod, and the side chains extend outward in a helical array. The alpha helix is stabilized by hydrogen bonds between the NH and CO groups of the main chain. In particular, the CO group of each amino acid forms a hydrogen bond with the NH group of the amino acid that is situated 4 residues ahead in the sequence. Thus, except for amino acids near the ends of an alpha helix, all the main chain CO and NH groups are hydrogen bonded. Each residue is related to the next one by a rise of 1.5 angstrom along the helix axis and a rotation of 100 degrees, which gives 3.6 amino acid residues per turn of helix. Thus, amino acids spaced 3 and 4 apart in the sequence are spatially quite close to one another in an alpha helix. In contrast, amino acids 2 apart in the sequence are situated on opposite sides of the helix and so are unlikely to make contact. The pitch of the alpha helix, which is equated to the product of the translation 1.4 angstrom, and the number of residues per turn 3.6 is 5.4 angstrom. The screw ends of a helix can be right-handed clockwise or left-handed counterclockwise. Essentially, all alpha helices found in proteins are right-handed. In schematic diagrams of proteins, alpha helices are depicted as twisted ribbons or rods. The alpha helical content of proteins ranges widely from nearly none to almost 100%. For example, about 75% of the residues in ferritin, a protein that helps store iron, are in alpha helices. Single alpha helices are usually less than 45 angstrom long. However, 2 or more alpha helices can entwine to form a very stable structure which can have a length of 1000 angstrom, 100 nm or 100.1 micrometer or more. Such alpha helical coils are found in myosin and tropomyosin in muscle, in fibrin in blood clots, and in keratin in hair. The helical cables in these proteins serve a mechanical role in forming stiff bundles of fibers as in porcupine quills. The cytoskeleton and internal scaffolding of cells is so rich in so-called intermediate filaments, which also are 2-stranded alpha helical coils. Many proteins that span biological membranes also contain helix. The beta sheets. Pauling and Corey discovered another periodic structural motif, which they named the beta pleated sheet. The beta pleated sheet, or more simply the beta sheet, differs markedly from the rod-like beta helix. A polypeptide chain called a beta strand in a beta sheet is almost fully extended rather than being tightly coiled. As in the beta helix, a range of extended structures are sterically allowed. The distance between adjacent amino acids along a beta strand is approximately 3.5 angstrom. The side chains of adjacent amino acid point in opposite directions. A beta sheet is formed by linking 2 or more beta strands by hydrogen bonds. Adjacent chains in a beta sheet can run in opposite directions, antiparallel beta sheet, or in the same direction parallel beta sheet. In the antiparallel arrangement, the NH group and the CO group of each amino acid are respectively hydrogen bonded to the CO group and the NH group of a partner on the adjacent chain. In the parallel arrangement, the hydrogen bonding scheme is slightly more complicated. For each amino acid, the NH group is hydrogen bonded to the CO group of one amino acid on the adjacent strand, whereas the CO group is hydrogen bonded to the NH group on the amino acid 2 residues farther along the chain. Many strands, typically 4 or 5, but as many as 10 or more, can come together in beta sheets. Such beta sheets can be purely antiparallel, purely parallel, or mixed. The beta sheet is an important structural element in many proteins. For example, fatty acid binding proteins important for lipid metabolism are built almost entirely from beta sheets. Tertiary structure. Myoglobin the oxygen carrier in muscle is a single polypeptide chain of 153 amino acids. The capacity of myoglobin to bind oxygen depends on the presence of heme, a non-polypeptide prosthetic helper group consisting of protoporphyrin 9 and the central iron atom. Myoglobin is an extremely compact molecule. Its overall dimensions are 45 into 35 into 25 angstrom, an order of magnitude less than if it were fully stretched out. About 70% of the main chain is folded into 8 alpha helices, and much of the rest of the chain forms turns and loops between the helices. The folding of the main chain of myoglobin, like that of most other proteins, is complex and devoid of symmetry. The overuse of the polypeptide chain of a protein is referred to as its tertiary structure. A unifying principle referred to as its principle emerges from the distribution of side chains. The striking fact is that the interior consists almost entirely of nonpolar residues, such as leucine, valine, methionine, and phenylalanine. Charged residues such as aspartate, glutamate, lysine, and arginine are absent from the inside of myoglobin. The only polar residues inside are two histidine residues, which play critical roles in binding iron and oxygen. The outside of myoglobin, on the other hand, consists of both polar and non-polar residues. The space-filling model shows that there is very little empty space inside. A system is more thermodynamically stable when hydrophobic groups are clustered rather than extended into the aqueous surroundings. The polypeptide chain, therefore, folds so that its hydrophobic side chains are buried and its polar charge chains are on the surface. Many alpha helices and beta strands are amphipathic. That is, the alpha helix or beta strand has a hydrophobic phase, which points into the protein interior, and a more polar phase, which points into solution. The fate of the main chain accompanying the hydrophobic side chains is important too. An unpaired peptide NH or CO group markedly prefers water to nonpolar milieu. The secret of burying a segment of main chain in a hydrophobic environment is pairing all the NH and CO groups by hydrogen bonding. This pairing is neatly accomplished in an alpha helix or beta sheet. Van der Waals interactions between tightly packed hydrocarbon side chains also contribute to the stability of proteins. We can now understand why the set of 20 amino acids contains several that differ subtly in size and shape. They provide a palette from which to choose to fill the interior of a protein neatly and thereby maximize van der Waals interactions which require intimate contact. Designated B. Thus, the hemoglobin molecule exists as an alpha-2 beta-2 tetramer, right? And subtle changes in the arrangement of subunits within the hemoglobin molecules allows it to carry oxygen from the lungs to tissues with great efficiency. Domain structure. Some polypeptide chains fold into two or more compact regions that may be connected by a flexible segment of polypeptide chain, rather like pearls on a string. These compact globular units, called domains, range in size from 30 to 400 amino acid residues. For example, the extracellular part of CD4, the cell surface protein on external certain cells of the immune system to which the human immunodeficiency virus HIV attaches itself. The comprises 4 similar domains of approximately 100 amino acids each. Often proteins are found to have domains in common, even if the overall tertiary structure are different. Reverse turns. Most proteins have compact globular shapes requiring reversals in the direction of their polypeptide chains. Many of these reversals are accomplished by a common structural element called the reverse turn, also known as a beta turn or hairpin bend. In many reverse turns, the CO group of residues N of a polypeptide is hydrogen bonded to the NH group of residues N+3. This interaction stabilizes abrupt changes in direction of the polypeptide chain. In other cases, more elaborate structures are responsible for chain reversals. These structures are called loops or sometimes omega loops to suggest the overall shape, unlike alpha helices and beta strands. Loops do not have regular periodic structures. Nonetheless, loop structures are often rigid and well-defined. Turns and loops invariably lie on the surfaces of proteins and thus often participate in interactions between proteins and other molecules. The distribution of alpha helices, beta strands, and turns along a protein chain is often referred to as secondary structure. The next topic we're going to deal with is Ramachandran plot, which will be in the next lecture. This will be just a general mention here, and I'll be giving details of this in the next lecture. Okay, so G2 Ramachandran plot. The formation of alpha helix and beta strands is at the heart of folding process. We therefore need to understand the factor that influences the secondary structure. Let us begin by considering the range of backbone conformations that are accessible to protein. Examination of the geometry of the protein backbone reveals that the peptide bond is essentially planar. Thus, for a pair of amino acids linked by a peptide bond, 6 atoms lie in the same plane, the alpha carbon atom and CO group from the first amino acid and the NH group and alpha carbon atom from the second amino acid. The nature of this chemical bonding within a peptide explains its geometric preference. The peptide bond has considerable double bond character, which prevents rotation about this bond. The inability of the bond to rotate constrains the conformation of the peptide backbone and accounts for the bond's planarity. This double bond character is also expressed in the length and bond between the CO and NH groups. The SCN distance in a peptide bond is typically 1.32 angstrom, which is between the values expected for a CN single bond 1.4 angstrom and a C double bond and double bond 1.2